Welcome everyone. So today let's discuss the topic transcription. Now as we all know, what is transcription? The process of formation of RNA, okay, from a DNA template. So that particular process, that means what we have studied during central dogma, that is called transcription. Now here actually what happens, for transcription to take place, the enzyme RNA polymerase is an important factor that is required. A DNA template is necessary. 4 types of ribonucleotide triphosphates like ATP, GTP, CTP and UTP, because we know that in RNA instead of thymine uracil is present, isn't it? So UTP is necessary. And another thing, divalent metal ions, magnesium or manganese, act as a cofactor, is essential factor which is necessary for transcription process. Now, in prokaryotes, actually a single type of RNA polymerase is required, but in eukaryotes there are 3 different types of RNA polymerase: RNA pol 1, 2 and 3. The prokaryotic RNA polymerase, it has a sigma factor which is also known as the initiation factor. That means the factor which starts or begins the process of transcription, and a termination factor which stops the termination, that is called the rho factor. Now let us discuss the transcription in prokaryotes. Okay. Now this actually occurs within the cytoplasm with the help of DNA dependent RNA polymerase. Now in prokaryotes, the structural genes, we know that it is polycistronic, that means it can form more than one polypeptide chain and are continuous. Here 3 basic steps are involved. One is initiation, elongation and termination. In initiation, first of all, now you can see in this diagram, this is a DNA template, okay. These 2 are the DNA strands, it's a double helix. Now this is the RNA polymerase and this is the sigma factor, that is the initiation factor. Now during initiation what happens, this RNA polymerase, it recognizes a particular point in the template. Template means the particular strand of DNA which will help in transcription or transcribing the RNA. So that particular point is recognized, okay, and the promoter reaches that, it binds with the polymerase enzyme, the sigma factor, and then it starts the process of synthesis of RNA. Okay. Now as the RNA polymerase, in the presence of energy and magnesium, is added there, so what happened, the elongation process begins. The RNA polymerase strand gradually it starts elongation, and during this process what happened, the sigma factor is released, because this is only the initiation factor. So once the initiation is started, that means initiation has been done, synthesis is started, then we can just simply remove the sigma factor. So the RNA polymerase now takes over the whole function and it will start synthesizing the RNA strand. Okay. Now it will continue till it reaches the terminator. The third step is the termination process. Here what happened, the RNA polymerase reaches the terminator region, which it gets attached to the rho factor. Rho factor is considered as the terminator factor, termination factor, and this factor will now just pull the polymerase out of the DNA strand, and when it is removed, the RNA is also released. That means the RNA synthesis is completed and the polymerase is set free to again bind to a next part for transcription process. Okay. So these are the basic main steps: initiation, elongation and termination in case of transcription in prokaryotes. In eukaryotes what happened, the transcription process occurs in nucleus and in 2 types of semi-autonomous cell organelles. We know that in plants 2 semi-autonomous cell organelles are present, mitochondria and plastid. There also transcription goes on. Now here the RNA polymerase 1, it actually transcribes the rRNA, ribosomal RNA, RNA polymerase 2 transcribes the mRNA, and RNA polymerase 3, it transcribes the tRNA, 5' RNA and snRNA. Okay. Now this is monocistronic, that means it has only one gene. The initiation of transcription, it requires an initiation factor. Here sigma factor or primers are actually absent in case of eukaryotes, that which we see in case of prokaryotes, and the newly formed RNA is synthesized by RNA polymerase 2, and this is called the primary transcript. Okay, it contains some unwanted base sequence which are alternated by some useful base sequence. Now you can see in this diagram, this is the RNA that is being synthesized, and here I am showing here some dark regions and some light regions. The dark regions are the useful parts which are called the exons, and the light regions are called introns, which are the unwanted base sequences. Okay, and so the primary transcript, it gets converted to a functional mRNA. This mRNA is first formed, that is not at all first, before it is not functional. Gradually it will become functional by 3 steps. One is the capping at 5' end. That means a cap is being added to the 5' end, which contains an unusual nucleotide methyl guanosine triphosphate. Now we are mentioning here MGPPP. Okay, so it is methyl guanosine triphosphate which is added to the 5' end, and that particular thing is known as the capping process. Now this cap, it actually protects the mRNA from degradation by nucleases and provides a ribosome binding site. Okay, and second process is the tailing process at the 3' end. This is also known as the polyadenylation. So this is the polyadenylation. Here what happened, a poly-A tail is added to the 3' end, okay, with the help of poly-A polymerase. Got it? And this actually contains adenate residues which protects the 3' end from degradation from exonuclease. Now the 5' end, here the cap protects the degradation by nuclease enzyme, and 3' tail end is protected from the exonuclease activity. Then third point is the splicing part. It is actually the removal of introns. Now we have already mentioned that introns are the unwanted base sequence. So these base sequence are to be removed. So how they are removed? By splicing process. Okay. And as introns are removed, then what happens? The exons will come close together and they will then become finally continual. The introns are removed with the help of S and RNPs. Okay, and these are formed actually by association of protein with the small nuclear RNAs. The end of the adjacent exons are then joined with the help of ligase enzyme. Now you can see here the small balls. That means this is the process of splicing where the introns are being removed, and when these introns are removed, finally here the exons are joined together, end to end they are joined together with the help of ligase enzyme. Okay, and finally what happened? A fully processed mRNA is released from the nucleus to cytoplasm for translation. Now this mRNA finally, it consists of the cap and the tail, and this is the functional fully processed mRNA which we required for the further process of translation, that is protein synthesis. So these are all the major events during the transcription process, that is basically initiation, elongation and termination factor, which is present in case of prokaryotes, but in eukaryotes no such initiation factor is there, but the RNA which is formed, that is which is called the first primary transcript, it consists of some wanted and unwanted base sequence. The unwanted base sequence introns are removed, that is called splicing. A cap is being added to the 5' end and a tail to the 3' end. These cap and tail protects the RNA from getting degradation by enzymes. So these are the important events, and finally the mRNA is being formed, functional mRNA. Okay, so the transcription process completes. So go through the videos properly, the notes are provided, go through them. If you find any problem, then please let me know.